So now we're going to switch into something completely different, which is talking about using epigenetic data. And and in this talk in particular, we're really thinking about ways in which epigenetic data might be particularly useful for economists. And I also want to acknowledge Jason and Shubha Basu, our postdoc, for some of the ideas that I'm going to be presenting today. We're hoping to put this into a paper, but these are kind of some early thoughts, so looking forward to your feedback there. All right, so just to give you an overview, I'll talk about what is epigenetics. And in particular, we'll talk about one particular epigenetic modification, which is DNA methylation. That's the type of epigenetic modification that you're going to find in most population-based studies. So we'll talk about its function and how it's quantified. Then I'll discuss some promises for epigenetics, also some huge pitfalls and challenges that the literature is working through. And then we'll talk about some open questions in the field. So first of all, what does the epigenome do? So it turns out we have this huge DNA packaging problem. We have to fit 5 feet of DNA, if you think about that, into a nucleus with a diameter of 6 micrometers, which is 6 millionths of a meter. So it's been compared to fitting Mount Everest into a grain of rice. If you can think about that, this is an incredible thing that our cells are doing in our body right now. So here you can just see in this kind of picture, we have our DNA kind of in its heterochromatin form inside the nucleus of a cell. It's wrapped really tightly around these chromatin fibers, so it fits in there. If we unravel the DNA, you can see kind of a picture of what we're more used to seeing when we look at DNA. And so this is part of what the epigenome is doing, is actually making sure that the DNA is properly packaged into the cell, into the nucleus of the cell. So it's maintaining this proper packaging of the genome across cell division cycles. It also regulates gene expression or transcription. So when the DNA is in this really tightly compact form like this, the transcriptional machinery can't come onto the DNA, read the DNA, and create an RNA transcript that can then go out into the cell and get translated into a protein. So the DNA has to be in this open form for it to actually be transcribed and read. So what the epigenome is also doing is kind of opening up certain areas of the DNA, closing certain areas of the DNA, and those patterns are going to be different in every cell because the genes that are transcribed in a skin cell, for example, are going to be very different than the genes that are transcribed in a muscle cell. So it's because of that really going to be cell specific, which we'll talk more about. But this is a very important function of the epigenome, is in fact, it's regulating which genes are transcribed. As I mentioned earlier, we're going to be talking about DNA methylation, which is the most studied type of epigenetic regulation. But just so that you know, there are other types of epigenetic regulation that are also common. So like histone modifications. So here you can see these histone tails. They're also playing an important function. There's small RNAs that are actually wrapping themselves around the DNA, keeping things from being transcribed. But the one that we study the most and that we know the most about, I would say, is DNA methylation. All right. So formally, DNA methylation is just when a methyl group is added to a DNA molecule at a cytosine base. And typically, when it's located in a gene promoter, it represses gene transcription. So it acts as a dimmer, and it kind of it can upregulate transcription, but normally it's kind of dimming transcription, so it's keeping that gene from being expressed. In mammals, this is almost exclusively found in what's called a CpG dinucleotide or a CpG site. So this is just formally when a C nucleotide is followed by a G nucleotide in the 5' to 3' direction. So we read DNA from the 5' to 3' direction. This is just determined by this phosphate backbone. But the point I want to make here is just when I talk about a CpG site, what I mean is a C, a cytosine, then there's the phosphate backbone of the DNA, and then G is the guanine. So that's just what we mean there. And these sites are where the methyl groups actually will get attached in the DNA, and they tend to be in gene promoter regions. So some important characteristics of DNA methylation. It controls which genes are active, as I mentioned. It plays a role in several key cellular processes. So things like cell differentiation, aging, and cancer are huge when it comes to the epigenome. As I mentioned, it's kind of determining what if a cell is a skin cell or a muscle cell. So that's a really important function because obviously our DNA is the same in the nucleus of every cell. So what areas of the DNA get transcribed in each cell is important for its kind of identity. And then I'll talk a little bit about its role in aging as well. It is highly influenced by the environment, which is something I think that has always captured the imagination of social scientists in particular. But it's important to note that it's also under genomic control. So both genes and the environment contribute to these methyl marks being placed. It's, I think, of huge excitement to the biomedical community because it is modifiable. So, already they're developing drugs and so forth that could potentially unmethylate certain genes. This has huge interest in cancer research. And because it is not just influenced by the genome, it's not as sensitive to ancestral differences. So you typically see in epigenetics research more a pooling of different ancestries. So that's just a lot more common. And, some of the things I'll be showing you today, you just don't get those same kind of challenges that you do with when you're working with genetic DNA in terms of ancestry differences. And I meant to say too, but feel free to interrupt me if you have questions as we go along. So how is DNA methylation measured and quantified? In large population studies, they're going to use microarray data. So usually it's the methylation bead chip, the 450K. The most recent is the Epigenetic. So in these arrays, basically what they're doing is they're just using fluorescent probes to quantify the percent methylation at a given CpG site. The Epigenetic now captures around 850,000 CpG sites. So that's kind of the information space we're working with, is about 850,000 sites. So the common data output is going to be the percent of methylation, the percent of methylated cells at a CpG site. So typically what people use are these beta values, which again is just the percent of methylated cells over just the total methylated and unmethylated cells plus some sort of just standardizing factor. So this is already very different from genetic data, right? Because with genetic data, we're used to working with 0, 1, or 2 alleles at a given site. Here, we're looking at a continuous measure, which is percent of methylated cells at a given CpG site. Just to point out that there's a number of population studies now that have DNA methylation data, and that number is increasing, in part because it now is pretty affordable and feasible to measure DNA methylation data in large samples. So we have, the Health and Retirement Study just recently released DNA methylation data on around 4,000 people. They haven't released the CpG level data yet, but they have released other epigenetic clocks, which we'll talk about. And you can see a whole other host of studies. So I think it's around like $250 now to do epigenetic data assay in an individual. So, and that price, will continue to come down. This is obviously not an exhaustive list. So just to give you an idea, though, many of the studies that we like to work with already have DNA methylation data or will be collecting it soon. So promises. So this is, I think, where the talk gets kind of exciting before did you have a question? Yeah, Remy.
Yeah, does the beta change across organs within an individual? Because sometimes they mention that they look at blood cells or liver cells or things like that, or is it mostly uniform within a given subject?
Say that again. I missed the first part of the question. Sorry.
Does the beta change across organs within an individual?
Yes, it's going to change across tissue types.
Okay.
Yep. So the epigenome is going to be different in every different cell type in your body. because it's performing this function of basically determining that that cell is, a muscle cell or a skin cell.
Can I follow up on that question? So
Yeah.
all those samples you showed, are they always using blood?
Typically, yes. And that's a challenge we'll talk about with epigenetics, but typically, yes, they're using blood just because of it's just simpler to collect, yeah. Okay. So I think a huge promise of DNA methylation data is to create biomarkers of aging and disease. So basically, because of these regulatory properties of the epigenome, it's capturing all of these cellular programming changes that are happening or occurring because of disease that's happening in the body or because of a person's changing health status and things like that. So it could, or has the potential to be this one-stop shop for disease biomarkers in the sense that you could collect somebody's blood and then, based on that, maybe construct 100 different biomarkers for different diseases of aging or health. One really nice thing about DNA methylation as compared to genetics is the effect sizes are quite large. So you can construct composite biomarkers in much smaller samples and still be well powered to do your analyses. It may also, if we can construct these reliable biomarkers, and I'll talk just in a second about how reliable are these really, but if we can construct these reliable biomarkers using blood, it could potentially help reduce misclassification of disease and so forth, or reduce self-reporting bias. So one particular example of this that has been really promising in the literature that's emerged over the past 10 years is the epigenetic clock. How many, just to get a kind of temperature of the room, how many of you have heard of an epigenetic clock, have been curious to learn more? Okay. Basically, we know that DNA methylation exhibits really extremely precise transformations with age. So as we age, the kind of cellular proportions in our body change, immune functions change. There's so much that's going on as we're aging, and our methylome is changing along with that. So what this epigenetic clock is it's just a composite score of CPG sites that are highly associated with chronological age or with phenotypes of aging. It's typically built using machine learning methods. So this is where it's different or a departure from the polygenic score, is in this field of epigenetics, they're more apt to use machine learning methods. Formally, it's just a weighted linear average then of methylation levels of these CPG sites that were chosen from the machine learning algorithm. They're using machine learning to kind of pick CPG sites that are highly correlated with some sort of aging outcome. And then you can take those CPG sites and apply them to your sample, put them in the same algorithm, and then it's going to give you your biological age, typically. So you can then use that biological age and look at a person's chronological age, and then look at deviations between the two to see if a person is aging faster or slower biologically than they are from their chronological age. And this deviation, which is typically referred to as epigenetic age acceleration, has been highly associated with aging-related diseases and mortality in the literature. And in fact, it turns out to be a better mortality predictor than traditional risk factors or chronological age. So this is what's been exciting in the literature, is that it does seem to perform much better as a predictor of mortality than kind of any other standard phenotype of aging that we have in like the health and retirement study, for example. There's over 14 clocks that have been developed to date for adults. There are also pediatric or prenatal clocks, but I'll just talk about the ones in adults for now. What makes these different, again, is that they're constructed using slightly different phenotypes in different training samples and across different tissue types. So here I'm just going to show you a few that have been developed. The first clocks were developed by Horvath and Hanum back in 2013. These were trained on chronological age. These are kind of called now in the literature the first generation clocks. They were kind of the first pass. Some of them are cross-tissue clocks. Some of them are just in blood. And these are really, really great at predicting chronological age. But of course, then what became more and more important as the literature evolved was actually being able to more accurately predict mortality and biological age. So this is what these second generation clocks are better at doing, is actually predicting mortality risk or time to death. So these are like the Levine clocks and the Grimmage clocks, if you've heard of those. And then the more recent evolution has been what we'll call the third generation clocks, which have been constructed by Dan Belsky using the Dunedin study. So in that study, he actually has longitudinal epigenetic data. So he's looking at the pace of aging. So in these clocks, they're comparing CPG sites across individuals within a cross section. What you can then do with these pace of aging clocks is actually look at the rate of change across individuals who are all the same age. So those have also, been emerging as exciting new advances. So this is kind of just one example of how we can use epigenetic data to capture biomarkers of aging that, have kind of emerged, I think, as more promising than other biomarkers of aging that have come before it. So in principle, you could do this for a lot of different phenotypes. The other thing that we think that might be really exciting for economists to think about using DNA methylation data is as biomarkers of past exposures. So we all know that retrospective collection of exposure data is tricky. It's subject to recall bias, misclassification, and it's impossible to collect for some exposures, right? So most people don't know how much lead they've been exposed to or how much pollution they've been exposed to, things like that. And here, the epigenome holds a lot of promise as an exposure biomarker, especially compared to other omics data, because DNA methylation tends to be more chemically stable and it doesn't degrade easily with long-term storage. And so this is not the case with RNA or with metabolite data. So for example, people have long used metabolites, and especially cotinine, which is a metabolite that's present in the blood if you've been smoking cigarettes. And that is a good way to see if someone's a smoker, but those metabolites only linger in the blood for about a day. RNA, similarly, the transcript will be floating around in a cell, but it quickly gets degraded, and it's very hard to preserve RNA over long periods of time. There's a growing body of evidence that DNA methylation measured in blood and other tissues is associated with prior exposures. And I think this is something to get excited about, quite frankly. For some exposures, in fact, the association may be strong enough to build accurate biomarkers that can estimate timing and dosage effects. So I'm going to give you kind of a quintessential example from the literature. This is also the most well-studied and well-validated example, and that is for prenatal and adult smoking. So DNA methylation is an accurate predictor of prenatal smoking exposure in childhood and adulthood. And in fact, what they've shown now in the literature is that CPG sites that get tagged when your mother smokes, these like prenatal CPG site marks, are different from CPG sites that get tagged when you smoke personally in adulthood, and that these two things can be disentangled. So here I'm just showing a figure from a recent paper here that points this out a little bit more. There's about 568 loci CPG sites that are related to maternal prenatal smoking. About 19 that have been pinned down for like early childhood smoking exposure. And then about 2,623 from own personal smoking. And these are all very separate. So you can see whether, your mother basically smoked while you were in utero. This is, pretty striking, I think, and it tends to be pretty robust. So they see, obviously they see these marks in cord blood, right after the mother gives birth, but they tend to linger, they're seeing into adulthood. So this is something that, potentially one could use. If you have DNA methylation data on an individual, you could construct a biomarker for whether or not, probability that the mother basically smoked, that you were exposed to smoke in utero. Thought I saw a hand maybe, but not. Okay. They've also been able to distinguish between, in terms of own personal smoking, that DNA methylation levels in the blood will differ by whether you were a current smoker, a former smoker, or a never smoker. So again, the methylation patterns will be slightly different. So you can take someone's blood and then you can see if they used to be a smoker, if they're currently a smoker, or if they've never smoked. They've also now, in terms of kind of getting excited maybe about looking at even dosage, not just kind of states of smoking, but also how much you've been smoking. There have been quantitative DNA methylation scores that have been constructed that show promise. And in this case in particular, they've been constructed to estimate like time since quitting and also number of pack years smoked. I want to say that this is really like an exemplar case. And we know that smoking has really huge effects on the methylome. So this is something that, is quite robust and has been shown. It's unclear if this would translate as clearly or cleanly to other adult exposures or prenatal exposures. So this is a really active area of research, and it's something that, we can get excited about. But just so you know, so far, it's really only been robustly shown for smoking. And, I think a lot of this is that the data just haven't been there yet to test it for a lot of other biomarkers or a lot of other exposures. But that's now, gonna be something that's coming down the pipeline. Overall, from what I kind of saw where the literature currently is, it seems like there's stronger evidence for persistence of prenatal marks than own kind of marks in adulthood. So just as an example, there was an RCT that was done that showed that folic acid supplementation in utero, those marks were still apparent 50 years later in individuals in their blood. And the same, of course, for famine. We know from the Dutch hunger winters, the Quebec ice storm studies. So these are some other areas where we see this strong persistence of prenatal behavior, prenatal marks. Any questions about that before
I have one question, but this is more general. It's about, this is a markers, right? They're not mechanisms or anything like that. So the link from exposure to epigenome has been established, but we don't know epigenome to anything else, right?
Say that we don't know epigenome.
The fact of the epigenome changes on anything else. We don't know the second thing.
Correct, yeah. So that's like another active area of studying is what are the mechanisms. And so these are biomarkers. They're not necessarily causal, but they're biomarkers.
Is there any sort of analogous issue with populations that, is there a potential concern if these associations are from homogeneous samples? The same way we worry about it with genetic data.
Yeah, and I'll talk about that a little bit in the passes. In the slides that are coming is that I think that's something that just hasn't been robustly studied yet, is whether these biomarkers really perform differently across ancestral groups. That's a good question, yeah. Yes.
So are you, from what you were saying earlier, is it true that these epigenetic effects might seem more visible if they happen, say, in utero or very early on, or would they be also visible later on?
So in terms of whether they're put down in utero or whether they're put down in adulthood, yeah, so it seems like for certain, so smoking is the kind of the caveat to this, where we do see really strong marks from both smoking in adulthood and prenatal smoking. I think with some of these other exposures, like for example, diet and things like that, they're not finding that those marks linger quite as long in adulthood, like if people change their diet. But with prenatal marks, in general, they seem to be much stronger and more persistent. And, I have to kind of look at a lot of this more, but I think that this makes sense in the case that our epigenome is completely laid down in utero. So, we are forming as human beings inside our mother. All the cellular differentiation is occurring as we're growing and developing. And so our epigenome is also defining that cellular differentiation. So that it kind of makes sense that we would see the strength of these prenatal marks. And then, of course, the Barker hypothesis, all this research that's come, before we had molecular epigenetic data has also been showing that. All right.
We have around 6 minutes or.
Oh, really? Oh, wow. That went really fast. So now to just, the one thing, and this is kind of, I think something where there still needs to be a lot of work, and it may or may not be something that economists get as excited about, but is to elucidate mechanisms. So, we could look at DNA methylation as a potential mediator of environmental exposures. If we want to know kind of how the environment is actually affecting a trait or an outcome, it is, going to be potentially working through DNA methylation. We could also think of it as a modifier of genetic risk, or G by E, again, because it's affecting kind of whether or not that genetic effect is actually realized. So these are other ways in which we could actually go on a gene level and look at the methylation that's occurring within specific gene regions or things like that. So it could, give us some more mechanistic insights and potentially illuminate, what's going on behind the gene-environment interactions. All right, so some pitfalls. The first major one that's already been brought up is the tissue issue, as it's often called, which is that each cell or tissue type, so blood, saliva, muscle, has a different epigenome. And so because of this, the predictive accuracy of these biomarkers may vary in different cell or tissue types. And I think there's also a big question about whether epigenetic biomarkers that are constructed from blood, say, as like a surrogate tissue for the brain, are going to be as predictive. So, if we're interested in constructing a blood-based biomarker for Alzheimer's risk or something like that, the question is can that really work? Or would we really need to get brain tissue in order to really construct a robust biomarker? And these are things that are, again, openly kind of being explored. Many tissue samples are always going to be really difficult to obtain. So this is also an issue and something that, again, is active work in the field. The second pitfall is, so we've been talking a lot about GWAS today. There's also a parallel in the epigenetics work, which is EWAS. And here, typically what they're doing is they put the CPG site on the left-hand side. So they're looking at how some sort of trait, like you could say educational attainment, which SSGAC did a study of educational attainment on methylation, but you can also flip it, and they often do. So oftentimes the beta gets put on the right-hand side or the left-hand side, kind of depending on the underlying kind of theory about the process. But this is where I think EWAS gets really sticky and not as helpful because of issues of reverse causality and also omitted variable bias. Just because it could be, there's some sort of change that happens in the environment that might induce some sort of epigenetic change that causes some sort of disease or something like that. But then the disease itself can also induce epigenetic changes. So it's really hard to uncouple kind of what's going on here, and the causal chain is much more difficult to unravel. And so this is an area that just gets a lot trickier with epigenetics. And I think, again, is a way in which economists can contribute. Everything we've been discussing is providing some more clarity to how we think about these models. The third is just that methylation microarray data are really messy. With genotype data, we have these really discrete values. We have reasonable quality control cutoffs. DNA is really stable over the life course, and it doesn't vary across tissue types. So it's much easier to kind of measure and quantify. With DNA methylation, it gets much messier. It's a continuous measure. It's sensitive to underlying genetics, environment, and importantly, experimental conditions and batch effects in the lab. So there's a lot more QC that needs to be done to make sure that the data aren't being affected by batch effects and other things. And this is, again, also a really active area, is just even determining, what are the best QC procedures. The cutoffs are not always straightforward. And it's going to vary from lab to lab, oftentimes, what people think is the best thing to do. So it's really important that you work with a good lab if you're interested in collecting your own data. And the third, as I mentioned, is it is influenced by genetics. So there are SNPs that, in fact, they're called methylation quantitative trait loci. So they're actually influencing methylation levels. And so because of this, a lot of these DNA methylation signatures we see may vary by ancestry. And so the effects of environmental exposures can be influenced by underlying genotype. And I would just say overall that I think best practices for accounting for genetic heterogeneity is really still in its infancy again in this work. And so, something that I think we'll see more active kind of work coming out to show how best to deal with that as more and more data become available. So final slide, just again kind of wrapping up and recapping some open questions in the field is, what extent epigenetic processes are causally related to environmental exposures and disease in humans. There's a lot of study in animal models that are underway. Definitely a huge need for more longitudinal data in humans and more causal research designs. And this in particular, there's so little causal work in epigenetics. So this is a huge area where I think economists can really, really contribute. Can DNA methylation signatures be used to construct stable and accurate biomarkers of past exposures? It's been shown for smoking. Can we do it for other exposures? What is the half-life of these exposures? Do they have permanent effects or do they fade away? Again, these are kind of open questions. And finally, can we use epigenetics to construct reliable biomarkers for aging and disease and other tissues or cell types? How much does it matter if we only have blood versus brain tissue, et cetera? So these are all open questions, but again, also areas I think to get excited about that, potentially have some stable biomarkers of past exposures. It's already been shown that, we have pretty good biomarkers of aging using epigenetics. So already some areas where you can dive in and hopefully some other areas where we can continue to discuss as these conferences evolve. So thank you, and happy to answer any more questions if there's time, yeah.